Welcome to today's session. In this session, we will discuss about UV visible spectroscopy, and these are the various subtopics which we will try to cover in brief in today's session. The basic principle of UV visible spectroscopy is valence shell electronic transition. In this, there is a movement of an electron between energy levels within an atom or a molecule. So, when an electron absorbs or emits energy, often in the form of photons, it transitions from one energy level to the other, as we can see in this figure. So, when electrons in the valence shell absorb energy, they can move to the higher energy levels temporarily and subsequently, then they may return to lower energy. Now, coming to chromophores. So, these are the group of atoms within a molecule that are responsible for its color due to their ability to absorb certain wavelengths of light. So, these groups usually contain conjugated double bonds or aromatic rings, as we can see in this case. Fine. So, the example is benzene or various azo-dyes. Here we can see that as so when the double bonds are conjugated in a compound, lambda max is shifted to a longer wavelength. From 178, it is shifted to 227. Coming to auxochromes. Auxochromes are functional groups attached to a chromophore that modify the intensity and wavelength of absorption and enhancing the color molecule. So the example of auxochromes are hydroxyl, amino or methoxy groups. So as we can see, the effect of hydroxy and amino group on the lambda max. Now, what is the difference between auxochrome and chromophore? So, auxochrome is a group of atoms attached to a chromophore that modifies the ability of chromophore to absorb light. However, in case of chromophore, this atom or group is responsible for the color of a compound. So exochrome increases the color of a molecule and chromophore gives color to a molecule. This is the main difference between auxochrome and chromophore. Coming to spectral shifts. So spectral shifts, it refers to the changes in the absorption or emission spectra of a molecule. So this may be due to the solvent polarity or pH, or either it can be due to the interaction of other molecules. So if there is shift to the longer wavelength, we call it bathochromic shift or red shift. And if there is shift towards the this lower like smaller wavelength, we call it blue shift. If there is increase in the intensity, we call it hyperchromic, and if there is decrease in the intensity, we call it hypochromic shift. Right. Now coming to the solvent effect on absorption spectrum, let's consider an example of phenol. Phenol contains OH group. Fine. So when we put the phenol in a methanol, there is shifts to longer wavelength, also called as bathochromic shift. This is due to the hydrogen bonding. Another example is that of a Nile red. It is dye molecule. So there is a shift from 559 nanometers to 625 nanometers in highly polar solvents like water. And the factor responsible for this is solvatochromism. So we might get a different spectra in a different solvent based on the polarity and their interaction of the molecules with the solvent. Coming to Beer-Lambert law, so it is the relationship between the concentration of a substance in a solution and the absorption of light through that solution. So it states that the absorbance of a solution is directly proportional to the concentration of a absorbing substance and the path length, that is L. So higher the concentration, higher will be the absorbance. Here I have given you an example. Please go through it. Now, coming to the instrumentation part. Instrumentation part is very important, and the major um, components of this instrumentation are sources of radiation, which consist of tungsten lamps or mercury or xenon arc lamps. Then lasers, which are useful for delivering specific wavelengths with high density. Then wavelength selectors, which consist of prisms, monochromators, or interference filters, which enables selection of a single wavelengths from a range of wavelengths. Then sample cell consist of quartz cells or covets, and these are useful for holding our samples. Coming to the detectors, so these are the devices which converts optical signals into electrical signals. So some of the examples are photomultiplier tubes, which amplify photon signals for low light detection. Okay, then there is a silicon photodiodes, which convert light into current across the broad spectrum, and then there is charge coupled devices. The other detectors are photovoltaic cell or silicon photodiode. So the different detectors have can be used for different purpose. Now, coming to the last part, that is applications of UV visible spectroscopy. So UV plays an important role in our day-to-day life. Let it be in food industry, pharmaceutical industry. So here I have mentioned three major applications of UV. The first one is spectrophotometric titration. This is useful in determining the concentration of a substance such as vitamin C in a sample by titrating it with a solution of a known concentration of another substance such as iodine. The next application is single component analysis. So this is useful in the determination of protein concentration, in which the absorbance is directly proportional to the concentration of protein in the samples. And the third major application is multi-component analysis. This is very important to analyze the composition of pharmaceutical tablets, because they consist of several active ingredients and excipients. So this UV visible spectroscopy is used to analyze it. Fine. So this is in brief. I have discussed about the electronic transitions. I have discussed about the auxochromes. I have discussed about the chromophores. I have discussed about the instrumentation part and then applications in brief. So this session you can use to quickly revise your entire topic. Fine. So please let me know which UV visible spectrometer is available in your institute, and please like, share, and comment. Thanks.